Welcome everyone at the webinar Good Practice Recommendations for PGT. Professor Martien de Rijke will start with the introduction of the webinar.
Hello, good morning everybody. Welcome to this webinar and thank you for joining us. So there will be 4 different speakers, each covering a different paper. Now what is important is that if you have questions, please put them in the chat box. And so at the end, we will have certainly time enough for answering your questions. Next, please. So my name is Martien de Rijke. I'm from the PGT Center from the University Hospital in Brussels. Next, and on behalf of the 4 working groups, I'm going to present the first part of this webinar. Next, I'm starting with a definition of PGT. So PGT is a test performed on samples biopsied from oocytes or IVF embryos. And the aim is to test for single gene disorders in PGTM or for chromosomal numerical aberrations as in PGTA or for chromosomal structural aberrations in PGTSR. The definition PGT also includes the testing with HLA typing. And so the testing will allow to select and transfer those embryos which are unaffected for the condition under study. Next. A bit of history. So the first ASHE guidelines for PGT were published in 2005. And a first revision was published 6 years later. The original paper was then split over 4 papers, each covering a specific topic. And this global structure has now been retained in the new 2020 papers. A first paper is on the organization of PGT service, and then there are 3 technical papers, one on biopsy, one on the detection of PGTM, and then the last is on the detection of PGT-SR and PGT-A. Next slide. Now, an important difference between the previous papers and the 2020 update is the change from guidelines to recommendations. What is the difference? Well, guidelines are strongly evidence-based. One is starting with a literature search, and this will provide evidence for the statements in the guidelines. Recommendations are different. They are statements based on the opinions and a consensus of a group of experts. So the 2020 papers are recommendations, meaning that there was no literature study done. And this is also why you will find just a short list of references in the papers. Only references to other guidance documents have been included. Next. So the 4 papers of 2011 were updated by 4 working groups. And each member has written a dedicated section depending on his or her expertise. And this text was then discussed with the entire working group until consensus during many online meetings. The 4 drafts were then checked for consistency and they were published on the ASHRAE website for stakeholder review. The working groups then addressed all the comments of the reviewers, and the final version was approved by the EXCO and was by then ready for publication. Unfortunately, COVID-19 slowed down that very last step of publication, but finally all is well that ends well, and we have now the papers published in HR Open, and they're also available on the ASHRAE website, together with the stakeholder review report. Next. So we have made these papers as concise as possible, trying to avoid repetitions. The drawback of that is that we used many cross-references within the paper and also from one paper to another. So one very important recommendation is that whenever you consult the papers for a specific PGT issue, you always have to consult the 4 papers together. Now, not right after this webinar, but very soon, ASHRAE is going to launch the papers in a different format, brochures, or a format which is more like a chapter with a table of contents, in which in the brochure the cross-references have been removed and the information was duplicated. So there will be 5 sections now with biopsy, PGTM, PGTSR, and PGTA all separated. And this should allow easier use of the recommendations. Another tool that may be handy is the glossary, in which specific terminology is explained, and this glossary is provided as supplementary data. Next. Now I will switch to the first paper, which is on the organization of PGT service. And so in this first paper, it covers the basic requirements of an IVF PGT center, meaning lab infrastructure, equipment, materials, lab documentation, training, and personnel. It also covers patient selection, counseling, informed consent, the pre-examination process, the examination process, the cycle, and then the follow-up of the cycles, also follow-up of the pregnancies and the children, accreditation and quality management, and also the item of transport PGT is covered in this paper. Next. A new section in this paper is the section on reporting. It's a section that provides a set of recommendations on how to report, plus also a list of the minimum information that has to be included in both the preclinical workup report and in the clinical cycle report. And we think this should be very helpful, as so far there was no clear standard available for reporting in PGT. Next. Another highlight of the organization paper is an overview of the most critical steps in the PGT workflow, where it is recommended that labeling and sample ID should be confirmed by an independent observer. And so the first critical step is after embryo biopsy. Second is during sample collection. The third step is in case of cryopreservation following biopsy. Step 4 is for further embryo culture. Step 5 is when the genetic results are issued. Step 6 is at warming or thawing of embryos. And then finally, step 7 is at the level of embryo transfer. These are the major high-risk steps, but in the technical papers, you will find details of additional risk steps where again you will need to verify the sample ID and labeling. So, next slide. I now hand over to my colleague Georgia Kokali for the second paper on biopsy. Thank you.
Thank you very much, Martine. I'm going to talk to you about the ESRI PGT Consortium and SIG Embryology Good Practice Recommendations for Polar Body and Embryo Biopsy for PGT. This paper provides technical information for the most applied biopsy methods and collection of biopsy samples for genetic testing. Before going into the highlights of the paper, I would like to talk about the training for biopsy and the training for tubing. First of all, embryo biopsy laboratory should be supervised by a person with recognized expertise in clinical embryology and preferably also basic knowledge in medical genetics. For the training of biopsy, we have suggested two steps: the preclinical testing, where at least 50 oocytes and 50 embryos should be tested, should be biopsied for all steps. And the second step is the supervised clinical training, where if we have a practitioner with extensive experience with micromanipulation, we suggest at least an additional 20 oocytes or 20 embryos. And if the practitioner on the biopsy is without experience in macromanipulation, at least an additional 40 oocytes or 40 embryos. Now, for the training of biopsy, the person, when they are training, should be supervised by an experienced certified clinical embryologist or a specialist geneticist, and they should always evaluate amplification outcome and ensure the absence of exogenous DNA contamination. Next. Now, laboratory issues related to biopsy are all stated in the recommendation papers. We have embryology issues for insemination, where ICSI is the preferable method, setting up for biopsy, labeling and witnessing, and quality control. For the biopsy laboratory infrastructure, we advise that the embryology laboratory design should include a dedicated area for biopsy or a separate biopsy laboratory room. For the biopsy equipment, inverted microscope with heated stage 3-dimensional micromanipulators, microinjectors, antivibration pads, stereoscope, incubators, and lasers should be included. And the biopsy material should be capillaries, IVF certified dishes and mineral oil, buffered media, HIPPIs, GMOPs or other, and the micropipettes used should have different diameter, inner diameter, according to the stage of the biopsy. So we suggest 10 to 15 micrometers for polar body biopsy, 30 to 35 micrometers for cleavage stage biopsy, and 25 to 30 micrometers for blastocyst. Next. The biopsy procedure consists of two main steps: creating the zona opening and then removing polar bodies or embryonic cells, which is the actual biopsy procedure, and mostly used is the method of aspiration. Then fixing or on a slide or tubing the biopsied samples. Rebiopsy could be considered only in the case of failed, incomplete, or inconclusive genetic diagnosis. Next. Now, the choice of which method should be used remains among the protocols and which protocol depends on the laboratory policy. For the blastocyst biopsy, day 3 assisted hatching could be used, or day 4 assisted hatching could be used, and then allow time for trophectoderm cells to be herniated from the blastocyst. Same day hatching based method could be used, and then again allow time for trophectoderm cells to herniate, or to perform simultaneously zona opening and trophectoderm cells retrieval. Next. One of the highlights of the paper is the overview of the merits and the limitation of each blastocyst technique. Next. Now, for the issues related to the sample collection means to have the efficient transfer of biopsied cells to slides or reaction tubes, which is a very critical step towards the success of the PGT cycle. Laboratory infrastructure, the tubing area should be in a DNA-free environment and is advised to be performed in a dedicated area or a room in a close proximity to the biopsy area. For the tubing equipment, microcentrifuge, a stereoscope or inverted microscope is advised, but it is acceptable to transfer biopsied cells to tubes with or without microscopic visualization. The tubing procedure could be in PBS or lysis buffer, depending on the protocol requirements of the PGT center. A new pipette is advised and strongly recommended for each embryo to prevent DNA carryover into the tubes, and the minimal amount of medium co-transfer with the biopsied cells into the tube should stay to less than 1 microliter. Next. I would like to thank the biopsy group, which is my colleague Giovanni Coticcio, Fernando Bronet, Caterina Celebri, Danilo Cimadomo, Joanna Lis, Sofia Nunes, Ioannis Fontouris, Elena Zakharova, Verle Gusens, Nathalie, and Martin Beric. Next. Now I will hand you to my colleague Celine Moutou. She will talk to us about what you need to know about PGTM.
Hello, everybody. For the PGT, I'm Celine Moutou from Strasbourg, thanks. For the PGTM paper, I would like to highlight 6 points, which are in light blue in the table of contents on this slide. STRs in the single cell or few cell methyl chapter, the work practice controls for targeted amplification, the validation process for different testing strategies, specificities in the preclinical and clinical reports, and the special case. Next. A table is included that defines informativity for STR markers. A ranking for PGTM is done according to the informativity of the couple from fully informative to informative, different partially informative, also called semi or half informative, and non-informative configurations. A similar table was included for the SNPs. Next. It is important to include positive and negative work practice controls. When the variant detection is included in the test, for autosomal dominant disorder, high risk and low risk genotypes. Male and female controls should be added for X-linked disorders, and for autosomal recessive disorders, carrier and normal genotypes, and when available, homozygous or compound heterozygous genotypes. A minimum of 1 negative control per buffer is recommended to control for contamination during the sample collection. This is the IVF laboratory negative control. And a minimum of 1 negative control with amplification mixture to control for contamination during second up of the amplification reaction, and this is the laboratory negative control. Next. The validation assays depend on the strategy for PGTM. For targeted amplification, a validation is necessary for every new multiplex. The minimum number of samples of non-genotype tested, of markers to be used, and the criteria for validation are described. For whole genome amplification, strategies followed by targeted amplification, MDA and trophectoderm biopsy are preferred, but other validated approaches are acceptable. A first implementation has to be done for WGA, and the locus-specific downstream application only needs to be checked on WGA products. Finally, a global validation is needed for WGA followed by generic testing. There is no locus-specific development, but informativity testing still has to be done, and bioinformatics analysis is crucial. Next. The preclinical report should include the indication and the gene, the pathogenic variant using HGVS recommendation, the gene reference sequence, the genome build used, inheritance mode, data on markers used, results from informativity testing, and finally, the strategy that is chosen for PGT cycles with a limitation and an evaluation of the residual misdiagnosis risk. Next. Recommendations were added for specific case. For de novo mutation, where phasing is possible during or prior to PGD. Consanguineous families, where informativity is limited by shared haplotypes and contamination detection has to be reinforced. HLA typing, where highly multiplex protocols are needed with a high risk of recombination. Exclusion testing, and mitochondrial disorders, where an expression thresholds need to be established and enzymatic digestion is needed prior to PCR. Finally, specific recommendations are made for embryo status nomenclature in the clinical cycle report. Unaffected, carrier or affected for monogenic disorders, low risk or high risk for mitochondrial PGT, at risk or not at risk for exclusion testing, HLA compatible or not HLA compatible, or inconclusive or abnormal or aberrant for other results. Next. I would like to thank all the members of the PGTM group, Philippa, Jos, Eftiga, Carlos, Georgia, Daniela, Verly, Natalie, and Martin, and I would like to thank you for your attention.
Thank you, Martine, and welcome to everyone from Maastricht. It is my pleasure to address you today on behalf of the working group that drafted the fourth and last paper in this recommendation series. My co-chair, Carmen, and the other team members, Dimitra, Eftigia, Julia, Maria, Francesca, Veerle, Natalie, and Martine. So thank you all for your hard work, valuable contributions, and smooth collaborations. It was my pleasure. So let's now move on to the highlights of this paper. As the title already implies, is on the structural chromosomal aberrations, referred to as PGT-SR, and numerical chromosomal aberrations, referred to as PGT-A. So the techniques that are being discussed in this paper are FISH, array-CGH, NGS, and SNP array as techniques used for PGT-SR, and whole genome amplified based array-CGH and NGS for PGT-A. If I can have the next slide. So there are actually two subjects in this paper, PGT-SR and PGT-A. And for all the techniques used, we have listed a number of issues. These are the laboratory issues, laboratory infrastructure, equipment, and materials, work practice controls that I will come back to, pre-examination validation as well, preclinical workup and reports, risk assessment, and finally, limitations of the test. If I can have the next slide. So the importance of performing a preclinical validation has already been stressed by Celine, and this is of course the same for all techniques that are being used. So I will not elaborate on the negative controls because they have been addressed. So at the lower part of the slide, you can see the positive controls for PGT-SR, are usually not readily available, as you need an material of an unbalanced phenotype. So what could be used as positive control is previous unbalanced products of conception, if there is material, balanced or normal lymphocytes, and some techniques can make use of unbalanced embryos that come available during a PGT cycle. I can have the next. So for PGT-SR and PGT-A, when using ARI-CGH or NGS-based testing, it's important to test enough replicates to get a sense of the variability and use a wide range of euploid samples. So you have an impression of what the standard deviation for calling euploidy is in your platform. It's also important to start to test a wide range of aneuploid samples to likewise get an idea of trisomy and monosomy thresholds. And if you are planning to do to report on embryo on mosaicism, then you should also include in your validations a cell mixture model where you test different percentages of mosaic cell lines in order to set your chromosomal mosaicism threshold. If I can have the next. So speaking of indications for specific test, it was decided that this should remain within the scope of individual clinics. But as ASHRAE and as the working group, we felt that FISH is not recommended for PGT-A, as only a subset of chromosomes can be tested and better comprehensive molecular approaches are available today. So for FISH-based PGT-SR, it is acceptable to study rearrangements involving small fragments or subtelomeric regions of chromosomes that are difficult or impossible to detect using other methods, as they usually have a threshold of about 10 megabases. And that selection of embryos based on sex for social reasons is not acceptable. I can have the next, please. So inclusion for PGT-SR. PGT-SR is only recommended if the technique applied is able to detect all expected unbalanced forms of the chromosomal rearrangement. And usually this means that you have to be able to identify 3 out of 4 segments. So when comprehensive testing strategies are applied, it is acceptable to use the information on copy number of the non-indication chromosomes to refine your embryo transfer strategies. If we speak about inclusion for PGT-A, then it was felt that PGT-A remains heavily debated in clinical practice, but that the application has been reported for the following indications: advanced female maternal age, referred to as AMA, recurrent implantation failure, RAF, recurrent miscarriage, RM, where it needs to be noted that in the guideline published by ASHRAE, it was said that PGT-A for recurrent miscarriage without a genetic cause is not recommended. And the last indication reported is severe male factor. If I can have the next slide. So for counseling, we felt that it was necessary to pay more attention to this part, as PGT has become more complex over the years, both for professionals as well as for patients. So there is a big task, an important task for the counselors to well inform our patients. So for PGT-SR, it is important to discuss that the applied technology may not allow to discriminate between normal and balanced results. Of course, there are techniques out there today that can do this, but if your technique cannot, then it's important to inform your patients on this. The pros and cons of embryo sexing by FISH should be discussed, which are that all male embryos, affected or unaffected, will be discarded, and that carrier females cannot be distinguished from unaffected male embryos. The clinical significance of transferring mosaic embryos is currently unknown, and the center's policy about the identification and transfer of embryos with mosaicism when applied or segmental aneuploidy should be documented and shared with the patient. And finally, for PGT-SR, when combined with PGT-A, the policy of embryo ranking and transfer should be discussed with the patient. Can have the next. So for the preclinical workup report, it was felt that for PGT-SR, an overview of the most likely segregation products and their possible clinical implications should be included. And that for the clinical report in PGT-SR cases, embryos with no or inconclusive results are not recommended for transfer, and that rebiopsy to try and obtain a result may be an option if feasible. Next. So this is my final slide on this paper that highlights the strengths and limitations of the various techniques that are most widely used in PGT-SR and PGT-A. I will not lead you through, just tell you that in the columns you see the various techniques, which are FISH for PGT-SR and ARI-CGH and NGS for both PGT-SR and PGT-A. And that in the various rows, you see different parameters that have been evaluated. The number of chromosomes that can be detected, the resolution, the possibility to detect or not detect fluidity changes, what to do with no conclusive results, whether or not abnormalities are diagnosed, if there are mosaicism-related issues, the possibility to detect UPD, the risk of misdiagnosis, the impact of biopsy on the test result, whether it's possible to simultaneously detect chromosome copy number and single gene disorder, and finally, if the method at hand is capable of detecting the origin of aneuploidy. I think that was my last slide. Yes, so I'd like to hand over to the moderator for the Q&A session.
Thank you, Edith, and thank you to all the speakers for the very nice presentations. We will now read out some of the questions and ask the speakers to address them. So our first question, how do you make the analysis of PGT outcome? Is it completely automatic or is it done manually? It's a very broad question. I don't know who can answer that one.
I'm sorry, could you repeat the question? Sorry.
Yes, yes, of course. How do you make the analysis of PGT outcome? Is it completely automatic or artificial intelligence, or is it done manually?
So I guess that depends on the technique that you are using. For FISH, it's done manually, but for the comprehensive method, in general, part of it is automated. And my personal opinion is that in future we might be able to make use of AI as well.
I would like to add that there will always be a part, even in that future case, that there will always be a part manually, because there is always the validation of the results. So if there's a double check, there's one person and then a second person verifying the results. So if the first part is done artificially, then I think there will always be a manual part at the second person.
Okay, thank you. And there's a next question, I think, for Edith.
And the question is, why does fish-based PGTSR analysis not allow for a distinction between embryos with a normal or a balanced karyotype?
This is because the analysis is done on an interface level where you have a conglomerate of all the DNA that is in your cell. And if you have a balanced or a normal constitution, you have the same of every chromosome. So you have 2 copies of all your chromosomes. So on an interface level, on a global DNA level, they look the same.
Thank you, Edith.
Here is one for Georgia. The time between blastocyst biopsy and cryopreservation is very important. Is it recommended to cryopreserve them as soon as possible before re-expansion? And why do we cryopreserve the biopsied blastocysts as soon as possible after biopsy?
It's best to cryopreserve the blastocyst immediately after the biopsy because it is collapsed at this stage. And it's advised, and all the paperwork and the work done and published in the literature suggests that the best result for the survival afterwards are when you cryopreserve the blastocyst within 1 hour, within half an hour actually, of the biopsy procedure.
Okay, thank you.
One more for Georgia, I think, is why do we not biopsy embryos at day 4?
Okay, on the paper, we have stated the 4 alternative methods of biopsy. One of them is the cleavage stage, the other one is the spent liquid media, and the other one is the blastocentesis. In the paper, we have briefly discussed these methods, and because there are not a lot of evidence in the literature, we kept a small part of the paper dedicated to these 3 alternative techniques.
Thank you.
Maybe I have some for Martin. Does the shipping of the biopsy sample for analysis in another country affect the results? The change of temperature and pressure during the air transport?
Well, this is discussed in the organization paper in the section of transport PGT. In theory, this should not affect because the conditions should be chosen as such that there should be no negative impact of the transport. So very important is to run tests before starting with a clinical transport cycle, in which you not only test the efficiency of the courier service, but also very important how the sample, whether everything stays okay with the sample after transport, and amplification, for instance, is just as normal. Also control on contamination. So in our experience, we often have to do more than one trial to have the conditions optimal.
Thank you, Martin.
Okay.
Another very general question, maybe also answered by Martin, is can standard insemination rather than ICSI be done prior to PGT? What you did.
Yeah, that may be a bit a tricky question. It depends on what method you are using. As it seems that when you are using SDR marker analysis, the lysis procedure that is included there is capable of freeing the DNA from sperm. So in that case, IVF is not recommended. But if you use whole genome amplification method, It is generally thought that DNA from spermatozoa is not amplified. So in that case, you might use IVF fertilized embryos for PGT. If you are using a trifectoderm biopsy, then of course you have more cells, and it's less likely that you have contamination of sperm cells. Or if you use cleavage stage biopsy, you might choose to biopsy 2 instead of 1 blastomere.
Thank you.
Maybe another one for Edith. After internal validation with cell mixing experiments, how do we choose which are the best thresholds for mosaic classification?
Well, I think that all depends on the outcome of your tests and the resolution of the platform that you are using. Remind that if you take 5 cells, a 5-cell biopsy, then the minimum amount of chromosomal mosaicism that you can see is 1 out of 5, which is 20%. So if you say you're going to detect 10% mosaicism, then you need a biopsy that contains 10 cells, which usually is less. So be careful of what you say you can do, taking into account what you have tested, what the results are, and what the resolution of your platform is.
Thank you.
Another question for Georgia. How to distinguish polar body 1 and polar body 2 when they are biopsied simultaneously?
Okay, this is a very technical question. Polar body 2 is round and smaller, whereas polar body 1 at the time of the simultaneous biopsy is more elongated. Also, the second polar body 2, is very attached to the cytoplasm of the oocyte. To avoid, if you're not very experienced, to avoid not being able to distinguish which polar body is which, in that sense, you should start using the 2-step polar body biopsy.
Thank you, Georgia.
I have a question for Celine. What's Escher's opinion about transgeneration PGD? So no transfer of female carriers of XL recessive monogenic disorders. How does Escher define which diseases are X-linked recessive or X-linked dominant? Celine? Maybe Philippa can answer the question.
Philippa,
Do you hear me
no
now?
sound. Oh, yeah. Yes, Celine, we can hear you.
This is Celine. Do you hear me now?
Yes.
Yes.
Yeah. Okay. So I was saying that it depends of the local legislation. Some centers will transfer and some other will not because of the law. What has to be to be known is that if you do not transfer a carrier embryo, you have less embryos to transfer, even when it's allowed. So this is not a general answer, it's a local answer.
Okay, thank you.
Let me see. Yeah, another question for Georgia. If the embryo is removed from the zona and the embryologist tries to reinsert it, does that affect the results?
Well, it may do. you need to, this is again not so much in the recommendation. Of course, the more you handle the embryo, the more invasive the biopsy procedure is. But if you have no choice, and I guess you are talking now about the blastocyst or the cleavage stage. I'm sorry, what was the question? Was it about blastocyst on cleavage to try and fit it in, or on the blastocyst?
That was not specified, an embryo.
Okay, once you do a cleavage stage embryo and maybe an additional cell comes out, it is a good idea to try to insert it back in. If the blastocyst is gets totally hatched due to the biopsy procedure, you shouldn't bother try to put it back within the zona. You may cryopreserve it as a totally hatched blastocyst.
Okay, thank you.
I have a question for Edith again. What are the acceptable values for non-conclusive results following PGTA?
I guess the answer is that all depends on the local situation, because it is not up to ASHRAE to decide which embryo should be transferred, yes or no. I think the most important thing is that the results and the possible how do you say that? Complications or implications of transferring this embryo should be discussed with the patients, that you should be transparent about what you know and also what we don't know about these embryos.
Thank you.
A general question that can be answered maybe by Martin. If you cannot send the cells to the lab for testing, how long can you keep the biopsied cells in the freezer at -20 before you can send them to the diagnostic lab?
I would say here that the best thing to do is to not to keep them that long, to send them out as soon as possible. And I would stretch this period not longer than a week.
Okay, thank you.
Maybe, yeah, go ahead.
Me?
Yeah, I thought you.
Just add that, yeah, I think it's safer if you, for instance, if it's about a whole genome amplification step, that once they have been through that step, that is better to store than the whole genome amplified products before, for instance, proceeding then with a downstream application, a SNP array or NGS or whatever. So yeah, keep the time as short as possible. Okay?
Thank you, Martin.
Maybe another question for Martin. Many of the centers implement PGTA, even if the request is PGTM alone without indicative reasons. Does this follow your recommendations?
Well, it's not up to ASHRAE to really say that this should be done or that it can't be done, but we do have in the new recommendations advice on how to deal with PGTM and PGTA, because we see that indeed more and more centers are with the generic technologies are implementing the two together. Mm-hmm. And then what is important is that you have validated the combination, and that for instance, in counseling, that what Edith was answering for a different question, that you are very transparent to the couples, that you do the two, and that your embryo transfer ranking and transfer policy is very clear. Is before you start doing this, that this is that the policy is very clear and can be easily explained to patients. I think that is very important, and that is actually what we have tried to write down in the recommendations.
Exactly. Thank you.
Another question for Celine. What is the difference between allele drop-in and general contamination? And how do we know if there's an allele drop-in?
That's a very difficult question. A global contamination should show several peaks. For example, for if you run a PCR, an allele dropping would more be a single extra thing on your results.
Thank you, Celine.
Another one for Georgia. Concerning the best for embryo, which moment is the best to open the zona pellucida? Day 3, day 4, blastocyst hours before, or blastocyst at biopsy time?
Sorry, can you repeat the question, please?
Which moment is the best option for opening the zona pellucida?
Once we perform blastocyst biopsy. Okay, as a working group, we haven't made a recommendation on which we think it's safer. They're all used and they're all valid techniques. There hasn't been any publication where we could test on similar groups of embryos all of these techniques from, for example, the same lab, so that we know which one is safer. For somebody who is not very well trained, we think that the more simple way is to perform the hatching on day 3 or the hatching on day 5, but allow time before the actual biopsy. Simultaneous biopsy technique needs very good training. So if somebody is not very experienced, they shouldn't start with that. I mean, with the simultaneous biopsy method. They should first get the experience from the other 2 techniques.
Thank you.
Another question for you, Georgia. What are the recommendations regarding the diameter of the hole made in the zona pellucida in an assisted hatching on day 3 in order to perform biopsy on day 5?
I cannot exactly talk about the setting of the laser machine, because different lasers are set to different pulses. However, it should be on the low setting, and the hole should be kept quite small, not as we perform when we want to do assisted hatching on day 3. It should be relatively small, because as the blastocyst expands, this will end up getting much bigger.
Thank you.
And then I have another question for Edith. What are the recommendations on the technique for PGTA?
So as I try to explain in my slides, the only thing that we do not recommend is the use of fish for the reasons I mentioned. And otherwise, it's up to the clinics or to the centers to use the technique that they feel most comfortable with, but most importantly, that they have validated really well before clinical application.
Thank you.
A general question, but maybe also to be answered by Edith. What are the main causes of having a result showing complex abnormalities multiple times?
I don't. What is meant by multiple times?
Yeah, not specified.
Okay, so well, on the subject of a chaotic chromosomal constitution, that must be the result of a series of meiotic and/or mitotic division errors, so that the embryo ends up with various chromosomal abnormalities that differ from one cell to another. Yeah, as I said, I don't know exactly what is meant by repeatedly, so that I don't know how to answer.
Okay, that's fair. Thank you.
I have another question for Celine. Sorry, what are the commonest techniques used for PGTM? The most common techniques.
Oh, I don't have a recent overview of the most common techniques. Since a few years, it was the multiplex PCR techniques, but now I think more and more people use SNP array. NGS is just starting, but I don't have really new data on that. Maybe Martin should have more, but I'm not sure.
Well, the data collection is lagging behind, but from what we see from the online database is that the multiplex PCR with the STR markers is getting less and less used because people are moving for PGTM also to trophectoderm biopsy and whole genome amplification. And so either then the whole genome amplified products are used in a targeted PCR reaction or in SNP array. We also see that from the AQA for PGTM, that SNP array is more and more used, and then really recent also now NGS, but that's only starting.
Yes, that's what I was saying.
Yeah, thank you.
A question for Edith. How much time before transfer is recommended to the best to thaw biopsied blastocysts?
So usually so the question was, what is the optimal timing to transfer the blastocyst?
Yes, so if you thaw a biopsied blastocyst, how much time do you need to do it before you transfer it?
All right. I guess that again is something that you have to find out in your own laboratory. So from my personal experience speaking, we always wait at least 2 hours to let the blastocyst re-expand before we transfer. Maybe Georgia has something to add to that.
Yeah, the same goes in our laboratory. We wait for 2 hours before transfer.
Thank you.
One question also about the freezing, which we touched upon earlier. What will happen to the biopsy cells if they are kept more than 1 week in the freezer before sending them to the diagnostic lab? Will the results still be reliable of the genetic testing?
I guess what Martin already pointed out is that it's important to get your amplified DNA material as quickly as possible. So not keep it longer than necessary as a biopsy in the freezer, but do the whole genome amplification, and then there is ample time to store and keep your samples.
Okay, thank you.
question for Georgia and from people who just start up. So for beginners, how can the biopsy be practiced like on uncleaved oocytes or fragmented embryos? Are there any tips or tricks to achieve the biopsy training for beginners?
I'm sorry, can you repeat the question?
So if you're just starting to train for the biopsy, how can you practice it? Can it be done on uncleaved oocytes or fragmented embryos? Do you have any tips or tricks for beginners? How do you start to train?
When you use no, unfortunately, I'm not sure how to answer this question. So the question is not, Edith, you have an idea?
Well, I think it's difficult to get material to practice on, right? So it could be an idea to go somewhere and attend a course on biopsy, where you are able to train with animal mice embryos usually. And if you are not able to do that, then there is spare embryos that you can test with. But keep in mind that these are usually of lower quality, so they will probably have a less survival rate. So don't get disappointed by that. And keep in mind that these are embryos of lesser quality. So yes, attend a course or try to get informed consent of your IVF patients to have their spare embryos be used for quality applications.
These are both very good advices. It has to do a lot with the local regulations of the country. For example, in Greece, we can get consent from patients, unused embryos, not unused, frozen embryos that are not longer needed. For example, in the regulation of Greece, maybe after the consent of the couple can be used for our practice, but it has to do with how the country has the laws and regulations. So I guess what you have suggested about the on training on course training, specific training courses is a good idea.
Mm. And then maybe a last question. There were quite some questions, and we didn't really read them out loud, on mosaic embryo transfer. So maybe Martine or Edith can explain a bit why this was not very much in detail in the current paper.
I think
Oh. No, go ahead. Go ahead.
Yeah, I just wanted to add that one important reason why we really didn't go into details is that currently the clinical relevance outcome of transferring mosaic embryos remains a question mark. And so it's quite difficult to write down recommendations on that. Yeah, it remains very difficult. I think until we have much more knowledge, it remains as such.
Yeah.
What do you think, Edith?
No, I completely agree. That's exactly the reason why we didn't get into detail. But maybe I can add that we are engaging in a project to as ESHRE to find out the people's attitudes towards chromosomal mosaicism in embryos and what to do with them, and what the outcome is of mosaic embryos that have been transferred. I think that is really valuable data, that if ESHRE will be able to collect that and share that with the professionals in the field, that we can all benefit from that.
Yeah, that's a project in progress. I think maybe many of you have already participated in a survey on chromosomal mosaicism. And so this survey results, together with then literature, will be in a publication later this year, hopefully.
Hopefully. Okay, thank you very much for answering all these questions.
Okay, I will close this webinar by, first of all, thanking all the participants. Thank you for being with us. Thank you to the Ashley people, and thank you to the speakers, and of course a big thank you to all the people from the working groups. It was a very long way, but it was really nice to have these collaborations, all discussions online. Thank you so much for that. So now I would like to draw your attention where you can find the 4 papers. You can find them in a dedicated section on guidelines on the ASHRAE website. There you can also have the link at the left-hand side with the link to the stakeholder review reports. And then there's also the link to the papers in HR Open. And then soon to come is then the link with the brochure. So the different format in which there are no cross-references, and in which you can have the table of contents that should make the recommendations easier to use. So once again, thank you all, and maybe see you or hear you next time, maybe in the annual meeting. Thank you.
Bye-bye.
Bye.
Bye.
Thank you.
Bye bye.
Bye.